So in this video we will look at lots of different things, but we start with how do phagocytes know what to eat, what to take in through phagocytosis. This is a fundamental part of the innate immune system. How do they know? And this is what is encoded in their DNA from birth. This is really the essential thing. So what phagocytes can do is they can look for pathogen-associated molecular patterns. And these are, like, what is a molecular pattern? A molecular pattern is a structure in a protein or nucleic acid or something that is always seen in certain conditions. So a lot of these different structures only appear in bacteria, like peptidoglycan or lipopolysaccharide or bacterial flagella. Others only happen in viruses, like double-stranded RNA. If any RNA virus wants to replicate inside that cell, there has to be double-stranded RNA. And that's a dead giveaway that there's something terrible happening in that cell. So if our own cells can detect lipopolysaccharide or peptidoglycan or double-stranded RNA, they know now's the time to respond. And so they have receptors called pattern recognition receptors that recognize and bind to these things. So I think of this as like the same way we have receptors in our nasal epithelium that give us our sense of smell by binding to certain chemicals in the air. You know, those things can tell us if something's bad. We can detect rotting food. We can detect a corpse and know that that is terrible and we should avoid it. Or we can detect something good and know it's food using those receptors in our noses. And that's what pattern recognition receptors do for phagocytic cells and other parts of the innate immune system. And then that analogy, we can extend it and say, well, there are foods that don't smell like anything. So like a glass of sugar water has tons of energy in it. And if I drink it, I'll get that energy. Great. It doesn't smell like anything. And there are a lot of deadly toxins that don't have any odor. Likewise, there are a lot of things in pathogens that are not common to all pathogens, so we can't have built-in receptors for them. So those things are what the adaptive immune system is for. So one important group of pattern recognition receptors are the toll-like receptors. And I like these because they give a good example of how pattern recognition receptors work. So they are the main pattern recognition receptors, and we find them on phagocytic cells and any kind of cell that is likely to be exposed to pathogens. They are evolutionarily conserved. That is, insects have toll-like receptors. They're named for a gene in the fruit fly called toll. We use them slightly differently, but the point is that our common ancestor had these a long time ago, and we can't say that for anything in the adaptive immune system. Let's look through a list of these. So toll-like receptor 4 down here, which is found in the plasma membrane of a cell. It detects gram-negative bacterial cell walls, and specifically lipopolysaccharide. Toll-like receptor 9 would be inside a phagosome membrane, and it would detect bacterial and viral DNA. So this is a table, and I don't ask you to memorize tables, but on the next slide, we're going to look at the locations of toll-like receptor 4 and toll-like receptor 9 and look at the logic of their locations. So toll-like receptor 9 detects bacterial and viral DNA. And it's going to detect those things if they are inside a phagosome. So that's what this is showing. It's a receptor for those things. Toll-like receptor 4 is facing outwards from the plasma membrane, and it's going to detect lipopolysaccharide. Well, that kind of makes sense, because if there's bacterium out here that has lipopolysaccharide, it's out here. And the lipopolysaccharide is on its surface. So this receptor can detect it and know, oh, well, now we need to do phagocytosis. This receptor would never see bacterial DNA because it's inside the bacterial cells. Bacterial cells are happy out here. But once the bacterial cells have been brought into the phagosome, they get broken down and their DNA is released. And that's when toll-like receptor 9 might detect it. So that's kind of the idea here, is certain internal pathogen-associated molecular patterns would be detected inside the phagosome, and external ones would be detected on the outside of the cell. So that's pattern recognition receptors. The other way that phagocytes know what to eat is opsonization. So they have receptors for opsonins. These are things that make objects more tasty, so to speak. Both parts of the immune system can opsonize pathogens. So the complement system, for example, can be driven by either system. It doesn't really fit in either system, but it can be driven by the innate system, and it can opsonize pathogens. Antibodies are completely adaptive. They can opsonize pathogens. So in both cases, what that does is make pathogens more appealing to phagocytes. These natural killer cells I'm going to come back to at the end. So they're innate cells. It would be nice to talk about them here, but we really have to understand the adaptive immune system before we talk about how these work, because these sort of fill in situations where the adaptive immune system can't do what it normally does. Another innate thing is interferon. And interferon is a cool thing to know about. We can use it as we can use certain types of interferons as treatments for viruses, and we can think about what interferons do. They are innate signals. So they're released by cells if those cells know that they're infected by a virus. So toll-like receptor 3 could detect double-stranded RNA, and that is a dead giveaway that there is an RNA virus infecting this cell. So if toll-like receptor 3 detects that, it will initiate a signaling cascade that will lead to interferon production. This cell is infected by an RNA virus, so it is a virus factory. So it is going to release virions that will try to infect nearby cells. The cell can't stop that. It has already failed by the time it's detecting this. So what it can do is release interferon in the short amount of time it has before the virus takes over its translation completely. So it's going to release interferon, and nearby cells can detect the interferon, and if they sense this before they're taken over by the virus, they can shut down their own translation. They block protein synthesis. They certainly slow down viral replication, but they also slow down their own translation. What that does is prevent this from becoming a virus factory. So if interferon is released and detected, this one won't become a virus factory to begin with. So we can think of interferon as a way a virus factory tells other cells not to become virus factories. Super cool, right? Well, many viruses actively try to shut down this process. So there's that. But interferon tries to prevent viral replication, and the unintended result of that is malaise and pain. So there's some thinking that the first sense you have of a significant viral infection, such as influenza or COVID, is when you start to feel kind of sickly, before you get specific symptoms, when you just feel off, a little bit sore. That seems to be from huge amounts of interferon interfering with all of your tissues. So that's cool. Let's end this video here, and in the next one we will jump into adaptive immunity.